So, just to give you a little bit of a flavor in terms of a spatial omics technology, there are two main streams of a spatial technologies where one is imaging based. Essentially, we take the images just like small molecule fish to measure each individual transcripts, where it is and how many we have for in the image by taking a microscope image on the tissue slide. Another way is more similar to what Theo just mentioned, like sequencing-based processing, where we're basically doing an in situ sequencing. So the outputs are more like in a form of a FASTQ file for any other short read formats. Regardless of your measurement or technology, at the end of the day, we reduce everything to gene expression matrix and location matrix, where we can know how many genes we have or how many proteins we have, and where XY coordinates or XYZ coordinates, where everything is. So what it becomes actually for pre-processing is you take the images, you process the images so that it's smoothed, deconvoluted, so that we can identify individual dots over here or individual sequences. And then it also involves segmentations where we can identify structures or images of the cells so that we can quantify these transcripts or sequences that we can map it back to the cells or XY coordinates. And then we can also extract features on cell shapes or intensities of the transcripts, intensity of the negatives or control probes. But then also at the end of the day, I mentioned count matrices and location matrices are stored in a form of an Anndata. I'm not sure how many of you guys are familiar with Anndata or seurat objects. Awesome. Yeah, I think it's like half and half almost. Yeah, and that is more of a common format for any single cell or spatial data sets in Python. And Seurat's more on the familiar type of a single cell or spatial for if you're a R user. And because these data sets are complex, when you collaborate with other collaborators, we also tend to build interactive visualizer or image viewer so that we can always go back and forth these image level information, which I'm going to show you a little bit later. So I'm giving CoxMx SMI, which is an imaging-based transcriptomics, as an example. If you finish all the runnings and export everything, you will get these large directory of files. Of course, there are raw files where you store everything, including like C-stacked images and all the compressed spots. The codes are saved all the way down to seurat objects where you can just plug it into R and start using it. What we're more interested is in the middle. TileDB is a common format for images and image associated information files, which is commonly used for spatial. It's basically like easy to understand. The way to understand is it's more like a decoded spot files. It contains RNA, false code, negative probes, and segmentation maps separately. And basically, it's an ingredient for all of the Anndata files that we can generate. So in TileDB, we will have individual images stored in like field of view. So every single images that we took across the slides will be saved in a format of cell composite, which is basically an immunofluorescence to image to identify the cell boundaries and structures. Cell overlay images are segmentation outputs, and cell labels images are basically identification of each individual cells and compartment labels as well, where we can understand each XY coordinates on the. So these are some of the images that were stored in TileDB. There also CoxMx allows you to get decoded into the flat files as well, which is super handy to use. Basically, a spot file in CSV format ready to be used, polygons, expression matrix, XY coordinates, cell level metadata, and transcript XY locations. And these I will go over in depth what they are, how they look like, and how the data structure is formed in the demonstration. So just to give you a preview, flat files has a cell polygons file where you can understand the boundaries of the cells, expression where the XY coordinates, how many transcripts for each of the cells have. By transcript, it's basically each puncta. an FOB positions file where the XY in space, where these field of views or cells exist, and cell metadata file, just like how single cell analysis works. You can understand the intensity, width, height of the cells as a metadata or features the cells that would be relevant. Lastly, the transcript of like literal XY or locations of each individual transcripts. So, and we summarize these when it comes to Anndata, which is quite similar in seurat as well, but just in slightly different format. But the backbone of the data structure is very similar, where we can store spatial omics or single cell omics as well onto this specific data format. We typically care most about the X slot, which basically is a count matrix where you have cells by genes data matrix. Um, for typically antibody, it's more of continuous, antibody-based methods, it's more of a continuous number. For spatial transcriptomics, it's more a discrete count matrix, just like single cell or bulk RNA sequencing. Observation metadata or every metadata of the cells or by the cells are stored in ops slots where you can understand each row has an information about each cell. And variable metadata has information on the genes or probes. Lastly, the observation multi or like to the last observation multidimensional annotations. It doesn't use quite often, but sometimes we tend to store spatial information in here as well. And the rest of the information, just like UMAPs or any other processing or additional spatial information when you have a segmented masks, are all stored in unstructured data, which is available through scanpy, squidpy, or other spatial compatible packages. And then also we do, at the end of the day, we tend to build Explorer. I don't really go through the demo on how to build them, but I added one example of actual one of the data sets in our lab in like how the Explorer would look like. So we can also build a visualizer. This is specifically using a tool called Vitase, and we can process everything and load our H5AD files in a JSON format and start exploring the data sets by looking into specific panels of looking at how the gene expression varies across space. And you can even select specific regions in the UMAP and show where these expression or where these UMAPs, where these cells represent in actual space. So you can play around with different cell annotations and expression patterns and compare, generate plots through these Explorer formats. We can kind of go, I can share this notebook, collab notebook after the lecture, but just to go through how do you, if you were to given, like if you were to be given spatial omics data sets, how you would start processing or generating and data files and how to process them. So very first step with the spatial and any also single cell as well is to have all of the required packages. scanpy is a popular package for all the single cell analysis. squidpy is more gears towards image level analysis. So if you're doing spatial, I recommend doing both scanpy and squidpy packages downloaded. To and which I added a couple of like all the packages that we need, including plotting and some of the dependent packages for SquidPy and download the data files. So this is more to for the demonstration. I uploaded one of the example data sets that's available through NanoString CoxMx SMI. So you can download public data sets and um, get spatial omics datasets as well. For homeworks, we'll get our real astronauts PBMC datasets to play around with these. But regardless, we'll get the spot files or Anndata files so that we can load onto the scanpy or squidpy domain. So this is the flat file that I was talking about. First, we do have an expression. The main essential components are actually three, where we can have an expression level matrix, which is basically a reduction from a transcript file that I just briefly mentioned. So each of the rows will have cells, columns will have gene targets, and values will be counts or intensities of a protein or gene. And metadata is again will be in the ops column or ops M column for spatial that has a cell level metadata or annotations. FOB positions are basically a global layout of where everything is in XY coordinates. So we first load SquidPy and read these individual files in a form of a flat file. So we can assign the path if you have images to load. Counts, metadata, and FOB files are loaded in CSV. And when you load A data, basically you will see an data object with number of cells by number of features, and you will also in ops column you'll have these different metadata of the cells, including XY coordinates, but also like the informations on the cells, what are the intensities of these morphology markers, and what are the areas of the cells which FOBs is coming from. UNS and OPSM file slots do have spatial information. Once you run dimensional reductions, which I already ran briefly before the class, you will have dimensional reduction information stored in unstructured information as well. So just to look at inspect the data sets, you can just simply use A data, load the A data and look into the basic structures. You can also call like number of observations, number of variables in these kind of example plots to example codes to show how the like what are the number of genes, like what are the targets would look like, or what are the metadata would look like, and these informations. And we can also load the spatial coordinates of each individual cells by calling by going into the space opsam slots, as I mentioned. So typical pre-processing of a spatial or single cell workflows do involve normalization, dimensional reduction, and visualization over UMAP for cell typing and further annotations and feature extraction. This is a really this is worth another round of lectures, so I'll try to explain through and guide you through the homework too, which is more focused on single cell and spatial analysis. But how many have you done scan like single cell workflow or spatial? You typically normalize and log transfer them to understand the overall distribution of the cells, look into dimensional reductions so that we can look into thousands or 6,000 or whole transcriptome level information all in the same spots. And at the end of this process, you'll get UMAP where you can do unsupervised clustering, which I think a lot of you have heard about, to basically understand what are the cell types look like, how they are similar or different to each other. Cutting short, lastly, we can not only do single cell level processing, but we can always map all of these analysis back into the XY coordinates, where you can build spatial graph using spatial segment or spatial scatter. If you don't have a segmentation information, you will use spatial scatter to add to map it back to like by cell types or clusters or any information. You can even do cell metadata and plot it onto intensities as like PANS-K intensity, for example, over here. And these are some of the considerations that we questions to make sure you understood everything in this demonstration. Yes, yeah, the ops column has the metadata of this information on the cells, and OpsM is more on the spatial information on the cell. And this is more on the side, how, if you were to do analysis, how would the spatial and expression clusters would compare? Yeah, essentially, expression clusters through all the latent clustering or like PCA UMAP are basically looking at transcriptional information on like how expression-wise they're similar or different to each other. Whereas like spatial clusters are more on the region side. So, which I don't really cover here, but there are a few different ways to cluster by spatial. There are different statistics like Moran's I, Ripley's K to understand how closely, for example, these zero clusters are clustered together, or how far apart. What are the expressions, like spatial correlations of these cell 0 and cell 1 that are spatially quite distinct. So that's what we're still looking at, that is very distinct from the expression level analysis. And this is more of an open-ended, like what kind of biological patterns do you want to see or you might see with the spatial clusters of cells? Yeah, cell types that interact. Yeah, communications, we do see a lot. Yeah, domain level information so that we can now start to segment. This is the alveolar regions. These are like stromal, like muscle regions, and like tumor regions, non-tumor regions. Yeah, and there's also interaction and distinct patterns where we can understand, like even in the tumor biology, tumor cells and the invasive fronts of like or tumor immune infiltrations and these states. Yeah, it can also inform a lot in tissue functions as well. Like for example, lung over here, it's highly like spatially compartmentalized. You can see alveolar regions, you can see airways. Liver, it's very zonated where you can see different function, like different metabolic functions across the space. And you can also see, I mean, it depends on the context, but you can start to question how the immune infiltrations, the inflammation regions do invade to a different locations within the body and just limited by your knowledge in the biology. We also tend to define grids in squares or hexagons. Hexagons more common in like Visium platform. Rest of the platforms are mostly covered with square to define, or basically instead of like XY coordinates, you can also like you're still in XY coordinates but form a hexagon format so that we can kind of understand the interactions between the spots better, especially if you want to do like spot-based sequencing technologies. But also not only we organize the cells in a like grid for easy XY coordinate calling, we can also define the locations and neighborhoods of the cells by calling, um, or neighborhoods of the cells by taking a cell and taking a radius and calling, defining a neighborhood. You can also take delineate distances and look into how the cells are forming the neighborhood by looking into their distance. And you can also do KNN clustering of all of these regions and look into how the cells are forming, how, define the neighborhoods in different ways. So these are three most common way to define a cellular neighborhood when it comes to spatial analysis. And then once you define these regions, and given that you have like cell type informations or cell type annotated, you can start to look into in the regions or in these specific neighborhoods, how they are enriched, like what type of cell types or what type of states of cells are enriched by taking a neighborhood enrichment plots, but also project some of these neighborhoods back into the XY coordinates to understand how these neighborhoods are forming networks or what are the spatial variations across the brain. Like this is, for example, the brain where you can see specific or developing brain, I think, where you can see specific regions that gets turned on and off or changes across the space.